So actually, I just left that as the title because that's what I, said I was going to talk about, but I actually cut the actual tool out of this talk. So apologies. But Graham kind of talked about an open problem session last week, I think, if you were here. So I'm just going to talk about theory, motivated by other people's data. OK, right. So this talk is thanks to everybody for coming and for talking to me. And it's been great to talk to people and to meet new people. And thanks for organizing. Everything I'm going to be talking about is stuff I've been doing with Graham. OK, so going to quickly go over some biological motivational examples. I apologize to those of you who have seen bits of this before, but I'm going to try to get to the new things quickly. One set of fun to think about examples are the blood disorders in humans that are thought to be around because they provide some resistance to malaria, despite being bad for you sometimes. That's probably why we know about them, because there are things that are good for you in terms of malaria if you're heterozygous usually, but also bad for you, especially if you're homozygous, and things like that. Maybe the most common is sickle cell allele. It's a single base pair change that's present on 4 different haplotypes across Africa, and it kind of looks like that, right? And another example, there's a whole bunch of alleles that result in G6PD deficiency. There's like 130, there's 34 of them that are at, not just seen a couple of times, and so present at some frequency somewhere. And here's a really nice map from a recent review showing how these alleles cluster across space. Okay, and this example differs a lot from the previous one because that one was a single base mutation, and here there's something like 130 or 150 maybe different base pairs that you could get mutations at and produce this phenotype. Okay, so motivation is that we're seeing something to do with adaptation, and we've got some sort of clusters on a map. Okay, and then for another motivational example, sort of for the second half, comes from the desert in the southwest of the US and northern Mexico, where it's desert, so there's lots of rocks, and the rocks are different colors depending on where you are. In particular, there's in a lot of places really dark outcroppings of rocks that are caused by like old lava flows. And for instance, you can see one there. And I learned recently that, I like to use this slide to say that, look how patchy, this is. There's lots of patches of dark rock. And actually, if you go to these places, that's not maybe the color you'd actually see, I learned recently. But it's something like this. So mammalogists back in the 30s noticed that when you went collecting out here, the color of animals that you caught would often, match the substrate that they were living on. Like you'd be more likely to catch darker mice or darker rabbits if you were trapping on areas with dark rock than otherwise. And they sort of observed this in some like a dozen species, and it's been observed, at least hypothesized in reptiles also. And Michael Nachman and Hopi Hoekstra looked into one of these systems, the rock pocket mouse. Here are 2 individuals of the same species that were collected on, from different populations, this one dark and the other light. And they showed that at least, in this area, there's a single locus, there's a single allele of large effect segregating at the MCR1R locus that's providing a lot of this, that's causing most of the change in color. And so you have a picture where there's this one allele that makes mice darker that is beneficial in dark areas and deleterious elsewhere because, if you don't match the color of the substrate, then you get eaten by owls more often. And if you don't believe that's a strong selective force, then look at this picture. Okay, and this is also sort of an interesting example because they did further work and looked at other dark volcanic outcrops in other parts of the Southwest and found that, while the change in color, it was, heritable and everything, it wasn't being caused by the same MC1R allele. It's interesting because MC1R has been responsible for adaptive coloration changes in a whole bunch of other taxa, like including things like lizards. So we've got this adaptive coloration change being shared, the same basis shared between different species, but then also not between different populations of the same species, which is kind of interesting. Okay, so I think that's it for the motivation. Okay, so I'm trying to think about the geography of adaptation. Okay, so when you're looking at a species spread across a landscape, when do you get, when you're looking at a species spread across a landscape that's been presented with some evolutionary problem, when do you get, the populations in different part of the range, different parts of the range, coming up with the same answers to that evolutionary question, and maybe communicated by migration, and when do you get different areas coming up with their own answers? Okay, and sort of how often those two things happen tells us something about, well, first it tells us about, what fitness landscapes look like, right? Like, if local adaptation happens all the time and is very important, then that means there's not sort of a single adaptive peak, but there's many peaks that depend on where you are. Now, it tells us about constraint, because if, different regions independently come up with the same solution to an evolutionary problem, then that's pretty strong evidence that there aren't very many possible solutions to that problem. But we can only say that if we know that they were actually independent. And of course, things like speciation, right? Because something we know about speciation is that lack of gene flow and differentiation is important. So the main sort of summarizing quantity that I want to get at in different models is sort of what's the geographic resolution of adaptation, sort of over what scale do you get different solutions to the same problem, for instance. And so like, under what circumstances do different regions share solutions or do it independently? And also, it'd be really nice to know, like, how can we expect to tell which is happening? Like if there's been the same change in different places, how do we tell if that was independent or not? Okay, that's the motivational slide. So the rest of the talk, I'm going to talk about a homogeneous landscape and then a patchy landscape. And I almost, at the last minute, reversed the order of these two. For one thing, because some of you, this is familiar to many of you, and also partly because it overlaps to some degree with pictures that a lot of other people, like Rick and Jasmine's talk, for instance. So I'll try to go through this quickly and get to this part. And I'm not going to be, I wanted to put in a slide with references to everything that made us think about this stuff, and I only got one set of authors in there. Okay, so for the first part, just want to think about a homogeneous landscape. So there's a continuous species range, with constant population density. The environment has changed so that some set of alleles have gone from being deleterious to beneficial. And we're thinking of a large class of mutations that all do pretty much the same thing. And so when you look at a simulation, you see the different precancerous clusters. I mean the different adaptations occurring and spreading. Right, and Right, so I was going to make a joke that there, I'm done. Okay. No, I was going to make a joke that this had been a model of cancer, but since Rick and Jasmine did actually a much more complicated model, like looking at the appearance of subsequent mutations on this, then I switched it over to talking about organisms on a landscape. but that's not we just draw cartoons and they did much more serious things. But it means that a lot of what I'm doing should be maybe familiar. Okay, so one thing that we've added here is that in the picture that we want to think about is that some alleles are present as standing variation. And sort of to be able to have a picture that makes sense, we want to have them present as deleterious standing variation. And sort of the way that we think about that is that there's some point process of little clusters of mutants that had been deleterious and then after the environment changes become beneficial. Okay, so then after the environment changes, these and any new mutations fix locally or not. They have to escape demographic stochasticity with some probability, and this is sort of how I encode that probability, right? And then if they do, they spread radially with some speed. The dispersal distance times the square root of the beneficial selection coefficient. Sorry, I should explain notation, except it's on the next slide. Yeah, okay. So combining all these things, we can put down in sort of a space cross-time picture the Poisson process of mutations. So there's some new ones and some ones present at t equals 0 as standing variation. Those are different Poisson processes with different rates. And then we have to thin them by the chance that they escape demographic stochasticity. And then if they have appeared at some location, at a location that hasn't adapted yet, they spread locally. And then ones that appear in places that have already adapted, we don't care about because they're neutral at that point. Okay, so this is the cartoon, and it's all Poisson processes. So by drawing sort of inverse cones and doing computations with Poisson processes, we can compute whatever we want. Okay, so the way we wanted to find out to get some expressions for sort of order of magnitude on what spatial scale do you see independent adaptations. So one way of quantifying that is well, it's the width of these patches. So here's time and here's space, and you say that some point here, that a point here in time and space has been adapted if there was at least one successful beneficial mutation somewhere in this cone, because then that one would have spread to cover it. Okay, so by turning things around, you can argue that that a good summary of the spatial scale of this process is the diameter of one of these cones in space-time in which you expect to find one successful mutation. Okay, so that solves this equation in general, where lambda 0 is the density of standing variance, pi is 3.14, lambda is the influx of new successful mutations, and v is the rate at which the waves spread. Okay, and with no standing variation, you can write down a solution more easily. And sort of the main thing that we want to get out of this, I think, is that it depends pretty weakly on a lot of the parameters. This power 1/3 in 2 dimensions anyways means that to change the spatial scale of this by a factor of 10, you need to change the dispersal distance by a factor of 1,000. And you have to change the strength of the selection coefficient, so how adaptive the new mutation is, by a factor of 1,000,000, which is kind of surprising.
So S is SB.
Yeah, S is SB. And you can also compute things like how long do you have to wait until most places have got the adaptive mutation, which is a good thing to compute if you want to compare this to data to make sure that you're getting reasonable numbers. And the proportion of these patches that happen from standing variation versus new mutation. Okay. So any questions about this before I go plug in numbers? Yes?
Would you like to give some intuition for why it's a cube, or is it just purely?
Well, because it's space cross time.
Okay.
Yeah, because the relevant thing is, each individual times a generation is an opportunity for a beneficial mutation to appear. And so you just have to wait until you get a chunk of those the right size.
If it was a linear habitat, it would be the square root.
Yeah. That might explain it. Okay, I seem to recall that having explained it to me in the past. Okay, so if we make up some numbers, if we get some numbers from the literature for the sickle cell case and plug them in, now what we want to compare things to is sort of this pattern is, happening over a scale of like 1,000 kilometers. And if we plug in a couple of different parameters for how deleterious you want sickle cell to be in the absence of malaria, which, this isn't quite right because it's, oh no, never mind, that's okay. You get sort of in both cases, so this is the deleterious selection coefficient before malaria appeared. You get numbers really close to 1,000 kilometers, the right answer, kind of miraculously. And even though these are really close, the proportion that you expect to have come from standing variation has changed a lot. So we get pretty much the same picture, but a little bit of a different story for what happened. And this shouldn't be too surprising because you wouldn't really expect to be able to tell the difference between a new mutation happening and a standing variant that was around in only a few copies beforehand. But this is how it works out. And if we apply this to the G6PD example, here there's more variance. And so the sort of spatial scale over which you see these is smaller. Like 350 kilometers if you divide the total area by the number of variants and take the square root. And you could plug in various estimates for SB. So this is the advantage to having one of these G6PD variants. And the main difference is that the mutation rate is, well, maybe naively 150 times bigger because the target is bigger. Right, so that's what we observe, and we get something that's on the right order of magnitude, but maybe a little bit small, and a fair proportion of them coming from standing variation. And we have here like the time that you expect adaptation to take, and sort of these are smaller than that, which is the right thing. Okay, so this all sort of makes sense and makes us feel good. Yeah.
What about environmental heterogeneity? Doesn't that disturb your calculation?
Like
I didn't think
that it's not actually flat in here. Is that what you mean?
What?
Like that it's not actually flat in here. Is that what you mean?
For instance,
Yeah, probably. Yeah, I mean, really I just only believe this in terms of an order of magnitude calculation. And really we probably ought to put in, rare long distance dispersal. When you get, like Oscar was talking about. And when we did that for the first case in our paper, it didn't make a huge difference. It changed the numbers we got out a little bit for the typical length, but not as much as you might think. So I think it's a lot more interesting to think about in this case, is one aspect of this that we're totally not modeling is what happens when the different adaptations meet each other, right? And that depends on how they interact with each other. Like what happens to people who have two different G6PD mutations. And I don't actually know the answer to that, but I think it depends on which ones you have. And also, not all these mutations have the same selection coefficient. Some of them are much worse than others, yeah.
Related to that, even if they're the same, this length scale assumes, it's just looking at until the spread of each allele meets each other, right? Isn't there then going to be a coursing due to the genetic drift or the longer time scales?
Yeah, there'll be, the first thing that happens is they all mix together, right? And that's why I sort of estimated the characteristic length, but just by dividing the total area by the number of variants. But the rate at which you lose them due to drift is typically much longer. And yeah, I didn't, right, that would be another good calculation. Maybe the right model here involves these different variants that have some complicated set of fitness coefficients, depending on if you're homozygous or heterozygous, and whether you're in somewhere with malaria and what other variants are around, and you might have sort of turning over of different ones through time. Yeah. But this is maybe the first thing to do.
Getting back to the question about the heterogeneity, it just seemed to me that the heterogeneity associated with the intensity of, say, malaria in different regions can differ quite a bit. Okay,
Mm-hmm.
and so, and you get a pretty big difference in scale depending on your sort of assumption of selection coefficient. That's the deleterious selection coefficient, or is that both of them? Or
I was just changing so the beneficial selection coefficient was changing from 0.25 to 0.04.
Yeah.
And I was keeping the deleterious one fixed at 0.1.
So I guess what I was just wondering is, have you actually investigated whether if there is some variation, there's a bias in the direction of the overall, of using your sort of global approach to estimating the scale, right, that you're using, taking the whole area. You could say, okay, let's suppose that I have a mixture. Would I get, do I get a reasonable, do I get the mean, or do I get some kind of harmonic estimate, or what is the real estimate of the scale and selection coefficient in some composition of selection coefficient?
Oh, okay, right, so heterogeneous across the landscape.
Yeah.
Yeah. Well, okay, so the one thing that I know about that, I guess, is that if selection coefficient changes across the landscape instead of once it establishes, instead of a constant speed wave, you have a wave that whose speed changes depending on where it is. And you can, wave your hands and make the argument that still what matters is its sort of average speed, right? The average speed ought to determine the scale in which you see things. So it depends on how fine-grained the environmental variability is.
So you're saying it doesn't have much effect?
No, I think it could, but I wouldn't even know how to begin to, I guess. It sounds like a lot of work to try and start making a more realistic model of that, and it would be lots of fun.
Yeah, I was just interested in whether there was a systematic bias that one could get at with just a toy model.
What do you mean a bias?
Well, you're coming out with an estimate of the scale
Yeah.
and under certain assumptions. And if those are, if you have just even a toy mixture of those assumptions, you could see whether the estimate is systematically one way or the other when the variance increases.
Right, so I think if the speed that we use is the right average speed, and I guess I don't know what kind of average, then it should be unbiased, but I don't know what kind of average. Last one.
Did you say what triggers the change from deleterious to beneficial?
Malaria, which is thought to have happened when agriculture started having like ditches full of water around.
So you have a date that comes in all across?
What do you mean? Well, like malaria didn't, as far as I understand, we think that malaria didn't used to be endemic everywhere. And then people started being around ditches full of water more often. And then
I'm trying to understand these age estimates. Are they going with some historical event that can be independently dated?
Right,
Is that what
so
you're shooting for?
people have estimated by, counting number of mutations on nearby haplotypes or something. That's one of the ways that they get these, that the various G6PD alleles are between 40 and 400 generations old. And so sort of as a check, I've computed under this model, how long should you expect like the whole population to have some of these alleles? And depending on whether it's weak or strong, that should be 7 or 30 generations. And if these numbers were much larger than those numbers, that would mean we're not fitting the model very well.
Are we supposed to be puzzled that malaria has been around longer than 7 generations, longer than 32 generations?
No, what this is saying is that like the population, under this model, the population would have been, most everybody would have had a G6PD allele within 7 generations of the onset of malaria. That's what this number means. And then since then, it's had time, those alleles have had time to mix up. Yeah, sorry, that wasn't very clear. Okay, I'm going to move on to the next part now. Okay, so back to the mice. So here we want to ask sort of similar questions, but now the picture is that the landscape is not homogeneous, but there's these sort of big patches of environment where an allele that I'll usually call the dark allele is beneficial, and that allele is sort of deleterious elsewhere. Okay, so so there's patches of dark rock, and then surrounded by light substrate, and these are separated by some relatively large distance. And I guess I should have drawn this distance as larger relative to the sizes of these patches. And we have mostly the same parameters, so I'll explain them when they come up. And the two things, so what we want to do is think about, how long do you typically have to wait for, a population of light mice living on one of these patches to acquire a mutation to the dark allele and become adapted. And then given that a dark allele is fixed in one of these patches, how long would you have to wait for sort of rare migration, past all the owls and so forth, to get that allele to another patch? And, to compare those two timescales. And then also if allele has gone across by migration, what should that look like? Okay, so the first thing is easy to work out, okay? Naively, right, there's some number of individuals living on this patch, and so the mutational influx is the number of individuals times the mutation rate. And then each one of those mutations has some probability of establishing that, I'm always going to approximate by twice its selective advantage divided by the variance in offspring number. Okay, so overall that predicts that the rate of influx of sort of successful alleles would be the area of the patch times the density times the mutation rate times the probability of establishment. Okay, so actually the probability of establishment at some distance decays exponentially as you move away from the patch, but I did some, exact calculations. This is distance from the patch. This is probability of establishment of an allele given that it arose there. And what we want to do is integrate over this blue area. And we're approximating it by a step function. And so see, we do pretty good just taking this naive approximation. Much better than we actually know any of these other parameters. Okay, so now let's think about migration. So suppose that dark alleles have fixed locally in one of these patches. So there will be dark mice present outside of that patch at sort of migration selection balance. And we know, thanks to Monty and other folks, that the equilibrium frequency as a function of distance looks like this. It decays exponentially with rate equal to the dispersal distance divided by the square root of twice the selection against dark mice on a light background. And there's maybe a square root correction in 2 dimensions. And sort of here's what it looks like. This is from a simulation where the allele is beneficial in this little patch in between the dotted lines. The colored jiggly lines are time slices, and the black line is the average. And down here is a space cross time chunk of that, where the intensity of the red tells you the frequency of the allele. And I've highlighted, I've sort of picked some of these dark alleles that happen to get pretty far from the original patch and traced back their genealogy. So you can see how they, descended from somebody originally living in the patch where they're adapted. Okay, so what does this mean here? This is sort of an occupation density, right? If this tells us that we expect there to be 0.1 mice per generation in this field, what that really means is that there's 1 mouse every 10 generations. Or actually, since they show up in sort of families, there's correlation. It's more something like 10 mice every 100 generations. And so this is the process by which a new patch will get colonized. And so to understand that process of colonization, we need to understand those migrant families a bit more. I'm going to wave my hands quickly about this. So in the continuum limit, in the continuum limit, the descendants of any dark allele outside of a patch where the dark allele is beneficial looks like a branching process, a subcritical branching process. And its branching rate depends on how close it is to the patch, right? Because the closer it is, the fewer light mice it has to compete with, but it's still a branching process. And that's something we can compute things with, but would still rather it was a homogeneous branching process. So one thing we can do to get that is to draw a bigger circle of radius r0 around the original patch, and then just look at sort of the families of migrants that leave r0. Well, they leave r0 and might go back in. But what we do is we only consider the genealogy of individuals that have had an ancestor outside of R0 more recently than they have an ancestor inside of the original patch. Okay, so that decomposes the genealogy of these dark alleles into trees that originate with migrants moving outside of here. Okay, And then if we take R0 big enough, particularly since we're interested in the rare ones that make it to a new patch a fair distance away, we want to be able to approximate that by branching processes that just have constant growth rate of 1 minus s. Okay. And so so here it's sort of reiterating that, that we want to make the approximation that between patches, the sort of migrant families that we care about are like subcritical branching processes, relative growth rate 1 minus SD. So the chance that one of those lives for time t is e to the minus SD times t. And as Leah was reminding us yesterday, we know what one of those looks like conditional on survival. Okay? If a migrant family has lived for a relatively long amount of time t, then it looks like a single trunk that moves according to the spatial notion that spits off these subcritical branches. Okay, and sort of for a long time, you get the spatial motion that's always sort of surrounded by a little cluster of close relatives with random but fixed, random size but of a fixed distribution, at least asymptotically. Any questions about that? Okay, so that's the approximation we want to use. So then we use that picture to work out the rate of successful migrants from one patch to the other. The chance that one of these families hits the new patch multiplied by the chance that it establishes, given that it has. Okay, so I think, okay, yeah, all right. Okay. And then, the main missing ingredient there is that we need to know the outflux of these families, okay? Because we need to know the rate at which these families sort of leave R naught and then establish in the new patch. But it turns out we're helped by comparing to the thing that we already know, which is the occupation density of this process. So the occupation density, so Q was the mean frequency at migration, the mean occupation density at migration selection equilibrium of these families. And the migration rate is the probability of establishment. So the first thing is sort of the integral around the boundary of the outflux of migrant families, multiplied by the probability that one establishes in the new patch, which is the probability it arrives times the chance that it establishes given that it's there. The second thing, the occupation density, is the integral around the boundary times the outflux of migrant families times the occupation density here, which is the probability that it arrives times the occupation density given that it has. So that tells us that these two things are equal up to a constant, which is the ratio of the probability of establishment, given that a family has arrived, divided by the occupation time here, given that it has arrived. The occupation time here, given that it has arrived, and ignoring the fact that it's beneficial here. Okay, and this is sort of, this works if the separation is large. Okay, so we can use that to get an expression, and then we can compute some things. And here's a phase diagram. And the main thing that we get out of this is that it depends fairly weakly on most of the parameters except for the distance. Okay, there's a pretty sharp transition between when you get sharing of adaptations by migration and when you don't. That's relatively insensitive to many of the parameters.
This is, for these results, you would expect that a little bit of long-range migration would make a difference, right?
Yeah. I mean, if it's reasonably likely that one happens to land in a new patch, then,
Or even very unlikely, just because in this model it's exponentially unlikely with distance.
right, yeah,
It
so,
gets so unlikely that you look for anything else that could happen, tornado spreading you or something. Right?
yeah. Biologists would pick up trucks. So. Okay, right, so then I won't tell you how, but, you can work out the distribution of the haplotype that the migrant that you expect the migrant to have carried along with it. Because the length of the haplotype is just determined by how many generations it spent in transit, because each generation is a chance to whittle down that haplotype. And if we're approximating this transit as a Brownian motion, a killed Brownian motion, then you can look up explicit expressions for the distribution of the time it takes to get across, conditional on getting there before it's killed. And we get some more expressions. And we can plug in some parameters to these numbers and get some answers out, which tells us, so here I'm summarizing things by the chance that so given that one patch is already adapted, the chance that another patch at distance r has adapted is sort of this ratio of rates. And even if you put in a pretty wide range of selection coefficients and mutation rates, you get pretty robustly that you expect independent adaptation over scales larger than like tens or maybe hundreds of kilometers, and shared adaptations on smaller scales than that.
So Peter, does that mean to say adaptation by mutation before migration?
Yep.
Right.
Thank you. And given that two patches do share an adaptation by migration, the shared haplotype should be like 56 kilobases. Okay, so we can compute stuff, and this is mostly with Graham. It was fun talking to these other people. So I have some time for questions.
Yeah, what would be the effect of having a carrying capacity here? I'm asking because I can imagine that conditioning on surviving in that desert, you condition on having a relatively large number of individuals. If it would be a critical branching process, conditioning on surviving, I guess you go away from the absorbing boundary.
Wait, I'm not following. Say again.
If you condition on it having a few individuals having reached very far away,
Yeah.
you make it likely that there was like a huge number of individuals a medium distance away? No?
No. Subcritical branching process, conditional on survival, looks like just a single
Oh,
trunk
yeah,
with
that's a
a
good point. Yeah.
fixed number of individuals. But
What's the
yeah,
number
I
of
think that-
individuals there? Then does it depend on how far you're away from critical?
Yeah, it's always limited by 1 over the selective disadvantage. It never gets much above that, you're right.
Right. So when that is small, it hits the carrying capacity.
Yeah, so I'm not sure how to connect this to carrying capacity.
Yeah, carrying capacity I think of as a maximum density
Yeah.
of individuals you can have locally. And probably it's in the desert smaller than. Oh, it's all desert, right?
Yeah.
But you have some limiting carrying capacity, and to connect it to the carrying capacity, I would say, so carrying capacity times S, where S is the deleterious effect you enjoy in the area between the patches. If that is much smaller than 1, something different might happen. Because then the branching process approximation.
Oh, so do you mean if like, right, so I think you mean that if light mice get eaten so much in dark rocks, they just don't establish at all? Is that sort of the...
We
You're
can
the
talk.
opposite limit where it's not so bad to be.
Where you like fix in the desert, even if it's like Muller's ratchet.
But the light allele is better there than the dark one.
Exactly, but by chance, if carrying capacity is low, right, is this what you're trying to
Oh,
Yeah.
I see, right, yes, right. So if local drift, yeah. No, that's true. Local drift would cause more stochasticity that, like in doing the branching process approximation, and you're sort of assuming that you mostly outcross to the light individuals. And don't have a good intuition for whether that should change things a lot.
But it's actually, it's even hard to reach this regime than I think you're thinking, Oscar, because it's the total number of individuals alive at any time across all the space that's limited to not much more than 1 over the selected disadvantage. So in any local area, it would be even less, I guess.
Yeah, I mean, there's a, we can talk about that.
I guess this is all for dominant, or at least not completely recessive. I mean,
Yeah,
is
it's
that-
for not completely recessive, and it would be really interesting to look into that case. Because if it's completely recessive, then on the one hand, it has a harder time establishing, but on the other hand,
But
it
it's
can
neutral.
transit better.
Because
Yeah.
you're assuming it's always deployed and it's outcrossing, right? That's what you were saying.
Well, I mean,
And like,
really it's haploid and nobody has sex, but, Any other questions?
If you look into the other process, basically the invasion of the white into the black type areas, do your estimations make any sense that way, or is it really just the other way that you can base your estimates because you have the distance and you have probabilities, the added probabilities of moving from one patch to the other patch versus the other way around? The invasion is so common that you don't really see anything, or you don't really get anything out of it.
But what do you want to know about the light ones coming into the dark patch?
Yeah,
What do you want to know about it?
just in terms of like the frequencies, and if you base your estimates on that event, rather than, your story was basically all about the black ones making it over in the next black patch.
Yeah.
So considering that the white ones basically go into the black patches as well, and
But what we're interested in is how long, sort of, like, are you thinking of the case where the dark mice first are established, the dark is the ancestral, and then you're waiting for,
no.
let's talk about it in a
maybe
minute and
not.
let people go get coffee.
I mean, the other thing that can happen, apart from this migrant coming across, right, is the mice who are locally there who are like others adapting to the issue, which is that the completing two processes of waiting for a migrant versus the mice who are living on that dark patch that are the wrong colour, mutating and adapting independently. So I think we are accounting for both types of.
Okay, I think we're, time's up.
Good. I think actually we have one more talk before coffee, but